Here is a higher magnification image where nucleated cells form an almost continuous pavement of cells one deep. I'll first describe erythropoiesis, the lineage that gives rise to our red blood cells, those very important cells that transport oxygen and carbon dioxide throughout the body within blood vessels. There are a number of stages in erythropoiesis, and again, let me stress they arise from the erythrocyte progenitor cell. Have an admiring look at this image. I think bone marrow is one of the prettiest tissues to look at using standard stains under the light microscope. You can see some mature red blood cells. All those small, pink, reddish or orange stained structures are erythrocytes. You can see in some a central pale region, indicating the biconcave disc shape of these very special cells. They have matured and are now circulating in the bone marrow and will eventually leave the bone marrow to enter into the general circulation. They arise from the proerythroblast. This is the very first stage of the progenitor cell starting to form what would be the final erythrocyte. These proerythroblasts are large cells of up to about 20 micrometers in diameter. The end product is going to be the red blood cells you've just seen also in this image. They're much smaller cells, only about 6 to 8 micrometers in diameter. So, a proerythroblast is a large cell that is eventually going to go through the differentiation process and finally end up being just a little package of hemoglobin, which is essentially what a red blood cell is. The proerythroblast has a mild amount of basophilia. It's going to differentiate into what we call the basophilic erythroblast, which exhibits stronger basophilia than you can see in the proerythroblast. Remember, it's eventually going to be a red blood cell containing a whole lot of hemoglobin, the protein carrying oxygen. So, you would expect to have a cell that accumulates an enormous amount of rough or granular endoplasmic reticulum and ribosomes to make that protein hemoglobin. The accumulated endoplasmic reticulum and ribosomes takes up the basophilic stain. The cell has now a factory to make hemoglobin. The next stage is this polychromatophilic erythroblast. The cell looks a lot smaller, but there is a variation in their size, ranging from 10 to 12 micrometers at their very late stage, up to about 20 micrometers in diameter in their very early stage of differentiation. But look at the cytoplasm. It's sort of a lilac colour and is lighter because the cell has now gone through into the stage where, having developed the factory to make hemoglobin and therefore being basophilic, it actually manufactures the hemoglobin, and hemoglobin, being a protein, takes up the eosinophilic stain. So you have then a combination then of bluish tinge representing the stain of the endoplasmic reticulum and ribosomes, and you also have the reddish eosinophilic type tinge related to the production and accumulation of hemoglobin, and therefore it looks a lilac type colour. We call it polychromatophilic because polychromatic means many colours. The cell then passes to the stage we call an orthochromatophilic erythroblast, commonly called a normoblast. Orthochromatophilic means there's only one colour. And if you look at the cell, the cytoplasm has now more a pink to reddish tinge compared to these polychromatophilic erythroblasts here that have a lilac-coloured cytoplasm. The nucleus is very condensed now because the cell has become a lot smaller, on its final destination to be a red blood cell, only 6 to 8 micrometers in diameter. And the colour of the cytoplasm is more eosinophilic or reddish due to continual accumulation of the protein hemoglobin. Finally, that nucleus is going to be extruded from the cell, because as you recall, the erythrocyte has no nucleus. It's just a package of hemoglobin. Well, it then goes through the stage of being a polychromatophilic erythrocyte again. And finally, it goes on its final lineage differentiation to be a fully functional, mature erythrocyte. And the reason why it is now referred to as a polychromatophilic erythrocyte, or sometimes we refer this as being a reticulocyte, is because as the cell gets smaller, the cell still retains a little bit of the factory that synthesizes hemoglobin. The cell has lost its nucleus, which is why we call it a reticulocyte, as opposed to a normoblast you saw earlier, but because it has got a lot smaller, as I mentioned a moment ago, it has hemoglobin in it, which stains pink or reddish because it's a protein, but it also still has small traces of the protein factory and therefore traces of the blue stain. And that therefore gives the appearance of these cells as being polychromatic or a lilac colour. And finally, these erythrocytes are now fully mature and will be released into the circulation. They appear nice and rounded with a biconcave disc shape and now full of hemoglobin.